My name is To Ken Lam. I'm the Executive Director of the Local Community Engagement Office at NTU. Before we start, let me do a quick straw poll. Now, how many of you are visiting NTU for the first time? For those who are not NTU students. Okay, not bad, not bad. Just want to tell you a secret. Now, NTU actually is one of the top 15 most beautiful campuses in the world. Okay, and it was selected by Travel and Leisure magazine of American Express in 2012. So in Asia, there are only two: NTU and Peking University in China. So with such a beautiful campus, please come and visit often, because university is a public premise, public place. Anybody can come and visit. Okay. NTU has been doing very well in the past few years in terms of research as well as education and teaching. In fact, as you may know already, we are ranked number 11th in the world in QS World Ranking. This is the oldest ranking organization in the world. We are ranked 11th in the world, and also ranked number one in Asia. So we have done very well in all those areas, and as you know, university has got the third objective, which is social responsibility. So we want to do more in this area. We're doing a lot already, but we want to do more. So we try to actually engage our community, Singaporeans, as much as possible. And this Monthly Talk series, we call it Did You Know Monthly Talk series, is one of the things that we are doing. So we actually started in July, and this is the 4th edition. And we're doing on the last Friday of every month. But for November and December, we'll probably do it a bit earlier because of Christmas and so forth. In any case, the fact that you've already signed up, you're already in our mailing list, so we actually give you the information very soon for the next talk and the rest, and subsequent talks. So I look forward to your participation as much as you can. All right. Now, without further ado, let me first of all introduce to you the speaker of tonight's talk. By the way, the title of the talk is called New Drugs for Old Diseases, the Challenges of Overcoming Drug Resistance. And we have our speaker. He's none other than our Professor Peter Preiser over here. Now, Professor Preiser is the chair of School of Biological Sciences and the director of NTU Integrated Medical, Biological and Environmental Life Sciences at NTU Singapore. He obtained his PhD in biology from University of Delaware in USA in 1991. And after his postdoc and appointment at Worcester Foundation for Experimental Biology USA, Prof. Preiser joined London's National Institute for Medical Research as a senior research scientist. Now, in 2003, he joined NTU as an associate professor, and he was later promoted to full professor in the year 2009. Now, Professor Preiser majors in parasitic diseases such as malaria. He has extensive experience in malaria biology and developing quantitative proteomic approaches along with transcriptional profiling to understand complex biological processes in relation to host-parasite interaction. Now, without further ado, can we put our hands together to welcome our speaker, Professor Preiser?
Technology goes. No, it's Kanan. Well, thank you, Kanan. I hope this was the most complicated thing you would hear for this talk about what I really do. It's a great pleasure to see so many here tonight, that you made it on a Friday evening. And what I'd like to do today is really give you an idea about some of the challenges we face today as a world, but also about some of the solutions or some of the work that goes on at the university like NTU that helps drive or leads to new discoveries in invention. I think it's a long time that I have been nervous giving a talk, but this is about the most intimidating audience that I've had to face in many, many years. It's quite easy for me to give a research seminar to my research colleagues. It's very easy to give a talk to students because you know exactly what they want. They want the answer for the next exam. So it's very easy to go forward. But here, I really, when I was asked to do this, I said, now who is going to come? And I realized that I think we have an audience here from high school students to full professors and everything in between. So I have, quite a challenge. And the only time I was more intimidated when I had to give a talk was when I was asked to give a talk to my son's school class at 5th grade, so 10, 11 years old, when I was supposed to explain to them about what I do. And I can tell you something, kids, 10-year-old kids are vicious. If you don't get them entertained, you immediately know that they don't like what you're saying. So please be a little bit more kind to me. So what I'll try to do is I've structured the talk really in 4 different parts. I've given you a little bit of information about infectious disease, which is the topic, a bit of a historical perspective. I'm giving you a little bit of an idea about antimicrobial resistance. What are antimicrobials? Why do they work? About the problem of resistance and the challenges they face. I'm giving a little bit sort of an overview about drugs and drug discovery, because this is partly what we're looking at. And again, particularly with an idea of what a university can do in this space. And then I'm giving a few examples of research activities of different people here at NTU. And I'll mention them all as we go along. But what I would like to say is I want to acknowledge not just the PIs of the lab that have shared the data, but all the people that are behind it that have actually done the work. But if I would list them all out here, name them all, we would be done with just reading out the names. So I'll just want to make sure that everyone appreciates that actually everything that I show, that is like one little slide, often involves years of work that is behind it. So, technology. Oh, I need to turn this on as well. Right. So, first thing is are infectious diseases a problem in Singapore? And I mean, I came in 2003, and the first thing I encountered in Singapore was SARS. So, infectious diseases in Singapore can be a really big problem. But more recently, I've just this is actually clippings from local papers, just highlighting some of the issues that are sort of in the news in Singapore. And TB, influenza, hand, foot, and mouth disease. My son had 3 episodes of this, so I'm very familiar with this disease in particular. But even things like malaria, Okay, malaria, which has the ability to come back into Singapore if the conditions are right. So that's just to give you an idea. Yes, diseases like this are potentially important enough to make the news. But what I'm going to look at initially is to really sort of get your attention is a little bit of infectious disease in the context of history, and really sort of when from the perspective of an infectious disease, all stars align to create the perfect situation to wreak havoc. And the first one I'm starting off is a disease which is, well actually this is not a disease. This is the oriental rat flea. So it normally lives on rats. But when rats and humans are in close proximity, they jump over to the human and they bite them. And the reason why I put this up is because this little thing is responsible for the worst infectious disease disaster that has ever hit the world, or at least what we know about. What it did is it transmitted a disease which is very nicely described here by Giovanni Boccaccio, who lived in the 14th century in Italy. And I'm going to read this out because it is actually quite striking. So it talks, how many valiant men, how many fair ladies, breakfast with their kinfolks, and the same night supped with their ancestors in the next world. The conditions of the people was pitiable to behold. They thickened by the thousands daily, and died unattended and without help. Many died in the open street, others dying in their houses, made it known by the stench of their rotting bodies. Consecrated churchyards did not suffice for the burial of the vast multitude of bodies, which were heaped by the hundreds in vast trenches, like goods in a ship hold, and covered with little earth. That sounds quite dramatic. And this is what you can now still see about these when in sort of excavations. These graves that he describes were bodies were just dumped in because there was no one there anymore to bury them properly. In 1347 to 1315, in a 5-year span period, this disease hit Central Europe, and it killed 25 million people. Now, to put this in perspective, the total population of Central Europe at that point in time was 75 million. So it killed 1/3 of all people that lived at that time. The disease came by a ship, no one is quite clear, and hit first Sicily, and then it spread through the whole of Europe, causing havoc in the UK, in Central Europe, etc. Again, a description now from a more recent book which looked at the history of this disease is, again, dead littered the streets everywhere, cattle and livestock roamed the country unattended, brothers deserted brothers. What he's talking about is a disease, it's called the Black Death or the bubonic plague, and it hit Europe during that period and caused real havoc. And as I mentioned, the primary culprit of all this was the Oriental rat flea. So we still have wonderful records of, or leftovers, which are a direct effect because of this disease that hit the Europe in the 14th century. And this is one of them. This is in Siena. Any of you have ever been to Italy? Siena is one of the most beautiful cities, and that is the Duomo, the main cathedral in the town, and it's absolutely beautiful. When you look at Siena, you have this view. So this is where the Duomo is. So this is Siena. There's the main plaza. There's a famous horse race which they have around there every year. There is the Duomo. There is the Torre, the cathedral. This is the city hall. And then there is this structure back here that most people completely ignore. And I'm going to talk a little bit about this structure because it tells you something about impact of disease. So in the 14th century, Siena was one of the richest cities in the known Western world. It was also bigger than Paris. And it had this grand plan to build the largest cathedral ever made. So that's ambitious. Rich, it used to be quite common. You show off your wealth by building big churches. And so they wanted to build the biggest one. So here we are again. This is the arch that I just showed, this little construct. Here it is. You see it seems to be connected by something else here. And in it is a parking lot. And this is, what I said, a monument to infectious disease, because what Siena wanted to do was, this is the plan of the cathedral. It's huge, and so in 1347, when they had started building this church, so they had been building it already for nearly 10 years, the plague appeared in Northern Europe, and it hit Siena in 1348. Within 3 months, 2/3 of the population has died. So you've just gone from 100,000 to 33,000. You know, and by 3 months later, you could hardly find any person, wrote a reporter. It was followed up by by famine and another 2 incidents of the plague hitting, and the population was, by the end, there was hardly anyone left. So the population was, the cathedral the people had wanted to buy was way too big for the remaining population, and critically, most of the people had died that actually knew how to build some of these. So they just said, let's somehow figure out how to finish off what we already have. And this is what they did. If you think of the big plan was this was going to be the church. Starting here, this is the main entrance, this was going to be the main ship, and these are the wings. And what they actually ended up doing is, what we're seeing here are the wings. So because of the plague, Siena never recovered, and it got frozen in time. You ultimately, when you visit Siena now, it's a museum that tells you about life in the 14th century. So we have the advantage of being able to visit these sites because people got killed off during an infectious disease. Now you might think is, 600 years ago can't happen again today. Well, it can. So I'm giving you an example of what can happen not so ancient. Just about 100 years ago, we had another infectious disease outbreak. And this is described by Isaac Starr, who was a medical student at the University of Pennsylvania in 1918. Again, he describes the symptoms. As their lungs filled, the patients became short of breath and increasingly cyanotic. After gasping for several hours, they became delirious and incontinent, and many died, struggling to clear their airways of blood-tinged froth that sometimes gushed from their nose and mouth. It was a dreadful experience. Well, what does the disease that is described for is the flu. And a specific variant of the flu, the Spanish flu, which hit the world just at the end of World War I. It killed 675,000 people in America alone, 200,000 of which died in the month of October. Overall in the world, somewhere between 30 and 40 million people died of influenza in a 6-month period. And Leonard Crane asked in his books, The Ninth Day of Creation, what is today's world, or in today's world, stopping something like this happening again? And here's his kind of pessimistic answer. It seems nothing at all. I'm going to be a little bit more optimistic. There have been some things that have happened since 1918 and 19, which at least have stopped this happening until now. And this is just to give you an overview of some of the key things that have happened in humanity's ability to fight infectious disease. And I start off with Edward Jenner, who in 1907 actually showed that you can vaccinate people against a very severe disease, smallpox. By the 1900s, we had vaccines against other diseases, including tetanus, rabies, and typhoid fever. But only in 1900 did we actually know what caused some of these diseases, when Robert Koch showed that microorganisms are the cause of many of these infectious diseases. This was then followed in the 1930s by the discovery of penicillin and other antibiotics, and the start of really the ability to treat infectious disease with drugs. In 1950, we had then vaccines against TB, diphtheria, and so on. In 1952, the polio vaccine, which many of us have taken. And in 1977, the last case of smallpox was diagnosed. Since then, there has been no more cases of smallpox, and it's the only infectious disease of humans that has become officially eradicated. Now, in polio, in this year, there are only 3 countries in which polio is still present, and there is a hope that maybe polio will be the second disease that will be eliminated. So, looking at all these positive developments, vaccines, antimicrobials, etc., it was not surprising that in 1967, the Surgeon General of the United States, which is the highest medical official of the country, delivered this speech to the Congress where he stated, "It's time to close the book on infectious diseases. The war against pestilence is over." That was a profound and very optimistic statement. Well, 20 years later, a new Surgeon General said this. And that's what David Thatcher, who reported infectious disease arising peril, the death rates in the United States up 58% since 1980. So clearly, something had happened in these 20 years which really changed the perspective of how we needed to look at infectious disease. I think one of it is that we weren't able to develop vaccines just continuously, so a lot of diseases we couldn't develop a vaccine against. And the other is antimicrobial resistance. The resistance, the ability of pathogens, disease-causing organisms to not get killed by these drugs anymore. And this is just from the BBC, just really, I have been scrounging the web, so I'm acknowledging them here, that I have cut and pasted things out of the web throughout. But this was just last week, when it sort of a commentary, all you need to know about the antibiotic apocalypse. So the antibody apocalypse, it is a vision that we are approaching a world where antibiotics actually don't work anymore. And we are back to the ages before penicillin and antibiotics, so that people die of simple infectious diseases, which nowadays can be treated very easily still with antibiotics. And there's a lot in the news in this year about this. Drug-resistant superbugs may be more widespread than previously known. This relates to carbapenem resistance enterobacteria, which are lethal because if the antibiotics don't work anymore. And here in The Guardian, again last week, antibiotic apocalypse, doctors sound alarm over drug resistance. So it is clearly something which is now hitting the headlines. But it was something that we could see coming a few years ago or decades. Now, this has real impact. It has impact on people in Singapore, both in terms of health costs on humans if you can't treat them, but also economic costs. 35 to 50% of Singapore bacterial infections are already antibiotic resistant to some antibiotics, which means the cost of treating patients is now 50% higher for treating patients with hospital-acquired antibiotic resistance infections in Singapore. So one of the most likely places to pick up antibiotic-resistant bacteria is in hospitals, unfortunately. What is even more scary is when they talk about what the vision is going forward, when they talk about antimicrobial resistance is globally predicted to cause more deaths than cancers, with over 10 million annually by the year 2050, and cost the world up to $130 trillion. This is not billion, this is trillion. These are staggering sums of money and reflects a staggering amount of suffering. So that's sort of the perspective of an infectious disease. I hope I got everyone's attention. No one's nodding off yet. Good. So let's talk a little bit about antimicrobials and resistance. What is actually happening here? So what is an antimicrobial or antibiotics? It is a molecule, a chemical molecule, a chemical compound that specifically is able to affect a certain function in the microorganism that is essential for the microorganism to survive and grow. And what is also very critical is that this molecule doesn't affect us, because it's no good to have a drug which kills a microorganism and kills the patient as well. It's not considered good medical practice. So this is what the reason why this is possible is because they target functions in microorganisms that are unique to the microorganism. And I've just listed here a few. So microorganisms, bacteria in particular, make a certain type of cell wall, which we don't have, our cells don't have. This cell wall is essential for their survival, and if you disrupt the ability of a microorganism to synthesize, or bacteria to synthesize this, a microorganism can't grow. And penicillin is a drug which affects this. But there's also differences in the way bacteria make proteins, which allows it to be specifically targeted. There are also some differences in cell membrane and metabolic processes. So what these antibiotics do is they hit certain processes in the bacteria which are unique to the bacteria. So then we had drug, but what was noticed quite quickly is that drugs only work for a certain while. And in some cases, the introduction of the drug and the appearance of a first resistance took a decade, for example here, ampicillin, while in others, it only took a few months. If you now think of it from a drug company perspective, you just spend a lot of money developing a drug, you put it on the market, and a few months later it doesn't work anymore. It's quite discouraging. So how do these microorganisms acquire resistance? And what is listed here is a whole range of different mechanisms that they can do to cause resistance. And I want to highlight this creates obviously a challenge from a perspective of developing new drugs, because there are so many different ways the microorganism acquires resistance. So they do things like actually modify the drug. They chemically modify it so it doesn't work anymore. They actually break it down. They cause a mutation, a change in the way their enzymes work, so the drug doesn't work anymore. They pump the drug out. So there's a whole range of different mechanisms that these microorganisms have developed, and that is causing the problem. So when we now look at it from today's perspective, how do we deal with this antimicrobial resistance, with the challenge of this that we are facing? So we're dealing, first of all, with about just over 200 different infectious diseases that infect humans on a regular basis. Many of them have now developed mechanisms of resistance, and what we now need to do is we need to figure out, A, how to re-increase or re-establish the efficacy of certain antibiotics that we already have, how to develop new antibiotics, but also how to create antibiotics which make it more difficult for a pathogen to develop resistance to. And then it's a very important aspect is actually to also quickly diagnose resistance effectively so that you can adjust treatment more rapidly. So when you go to a doctor and it says, yes, you have this and this infection and you need to antibiotics, they also know this microbe you're infected with is already resistant to this, so we need to give you something else. So this is the history. Now I'm going to talk a little bit about drugs and drug development, and particularly the role a university like NTU can play in supporting drug development and discovery. And this is, again, something I got from the internet, but lots of these figures available out there is the normal drug development process. So what you start off with is some kind of drug discovery. You have a lot of compounds which seem to be able to do something to a pathogen. Then you evaluate whether these things are actually safe to give to a human and not in. So the preclinical to make them nice and good, and then you test them in humans, and eventually you get out a drug which is approved for use. This process takes 10, 12, 15 years depending. It also costs a lot of money. So this initial phase costs, takes a few years and can cost $10 to $70 million, and then you can add up a few 10 million, 50 million, 200 million, 500 million. So by the time you get a drug to market, you've spent between $500 to $1 billion. A university is particularly well suited to facilitate and enhance this process because of its huge resource of know-how that sits in different schools and departments. They can create an environment which is much more flexible and has a real fundamental knowledge of how maybe some targets can be identified, etc. What is also relevant, it is that industry today has relatively little interest in this part of the process because there's a lot of attrition. So as I said, you start off with 10,000 to end up with 1. So it becomes interested when you have something which is ready to be considered taken into human. So there is a greater interest to engage the pharmaceutical interest when universities play a role here and get to this stage. So this is sort of a little bit what is happening at the university, at NTU, just like at other universities. There is a basic biology which allows us to identify what is maybe a good target for a drug. There is the chemistry expertise which allows us to synthesize molecules which might actually inhibit these targets. There is the ability to do safety, medicinal, the study of how these drugs behave in a body, etc., before you go into human. This is all there. And here at NTU, we have particularly this expertise to drive forward these processes. So the last 10, 15 minutes, I will therefore now talk a little bit about 5 different projects from 5 different labs, from Kevin Petty's lab, from my own lab, from Kimberly's lab, Kimberly Kleinson's, Yang Liang's lab, and Julian Leska. And they all talk about different diseases where resistance has become a problem and different types of solutions of how solutions are either being conceived or actually have been found. So Kevin is an associate professor in the LKC School of Medicine, and he works on TB, which is a big problem. And he used a deductive approach to identify a new target and then develop a potential drug against. So just to give you a bit of an idea about TB, it's a very old disease. The agent is a bacterium called Mycobacterium tuberculosis. It usually infects the lungs. And there are studies that show that even in mummies 5,000 years old, they died of TB. So this bacterium has pretty much been around in humans forever. It was actually thought to be a disease which jumped from cows to humans, because cows also get a form of TB. But actually what recent work has shown, it's the wrong way around. It is actually humans gave cows TB. So not everything, humans have an impact on animals in quite surprising ways. So today, there are 2 billion people in the world, which is about a third of the world population, which are latently infected. What that means is they carry the bacterium, but they show no signs of the disease. In a small percentage of these people, the disease suddenly becomes active, and they get active TB, which happens in about 8.7 million cases every year, with about 1.4 million deaths each year. So this is a significant problem. What is even further a problem is the fact that we now have multi-drug resistant TB, which is resistant to the most potent drug, where you have up to 70% of treatment failures. And in 2015, there were 58,000 cases of this identified globally. While multi-drug resistance TB is rare in Singapore, there have been cases of this. One of them happened in Amokir, and there was the first case reported in February 2012, another case in 2012, and then a fifth case in 2016. And what was interesting is, while these are very few cases, the bacterium that caused these cases was always the same. So there is a hidden source of this bacterium in the population which can be reactivated and cause multi-drug resistant TB, even in Singapore. Now, when you're dealing with multi-drug resistant TB, you have a real challenge in terms of how do you treat it. It's already challenging to treat normal TB. When you have normal TB sensitive, you need to take 4 drugs for 2 months, followed by 2 more drugs for 4 months. You need to take drugs for 6 months in total. You can imagine the bucket of pills that you need to take home from the GP when you start your treatment. There's a problem that people feel after a while better and stop taking the drugs, and this is, causes potentially the risk of resistance developing, but also that taking some of these drugs for a very long time causes toxic effects. Now, in multi-drug resistance, this whole thing just gets exponentially worse. You now need to take 18 to 24 months of drugs daily. That's 14,000 pills and 6 to 12 months of injectable drugs. These drugs don't work though good. They are quite toxic, so they're not as nice. So people who get treated for multi-drug resistance are suffering. They're suffering because of the disease, and they're suffering because of the drugs. So how was TB treated before chemotherapy? Well, the only treatment that was really available was to tell people to go to high altitudes, because it was noted that in the high altitudes, somehow people staged a recovery. And this is just a picture of a sanatorium in Davos, which is an altitude of just over 1,500 meters. So this was how you treated TB before 1943, when streptomycin, an antibiotic, was discovered that worked against TB. So why did moving people to a high altitude actually reduce or lead to recovery? The reason for this is that, like humans, Mycobacterium tuberculosis requires oxygen. It's a strictly aerobic bacteria, so it requires oxygen to grow and survive. When you move up the mountain, the oxygen levels reduces, so you are ultimately starving the bacterium of oxygen. So the question therefore is can you use that information to figure out new ways of treating the disease? So why does mycobacterium require oxygen? And it's like human cells, we require oxygen as an oxidant to generate energy. So we ultimately burn food, and that burning of food generates energy, and in a cell, the energy that is generated is ATP. And this is critical for all processes. And for mycobacterium, having oxygen for this is essential. The process where ATP is generated with oxygen is called oxidative phosphorylation, and this process involves an enzymatic pathway that exists in mycobacterium. And I don't want to go into too much chemistry, but ultimately what happens is there's a number of processes, and at one critical stop, oxygen is required to generate water, and this then allows the generation of ATP. So the idea is there a way of blocking this in the mycobacterium so it can't make ATP? Because without ATP, it can't grow. And in Kevin Mthly's lab, they did actually find a very specific molecule, which is named Q2003, here it is, which is very good at blocking this step. So they actually went and found a target, and then they developed an inhibitor to this target, and they showed it actually worked. So when you treat bacterium with this compound, it can't make ATP anymore. And now I have turned the whole thing off. Okay, so this was very exciting, and it has actually been shown to be one of the most potent anti-TB drugs developed to date in terms of its efficacy. It does do exactly what it's expected to do. It works in animal models, and this inhibition of oxygen consumption actually kills multi-drug-resistant clinical isolates, and it is now in the later stage, or just about going into the second stage of human trials. So it is extremely positive and discovery, and so there may be actually in a few years' time a real new anti-TB drug on the market. Important, it's safe at this stage. Okay, now I talk a little bit about my own work in my own lab, so I'm feeling much more comfortable about this because I know it so much better. And is this approach that takes advantage of the fact that we are at a university, and not only do we work in biology and disease, we also have a very strong chemistry department. And this interplay allowed me to do something which I'll talk about here. But first, just to give you an idea about malaria, you know, it is still a global disease, and if you think of Singapore, it's surrounded by a sea of blue, which are the countries where malaria is still endemic. In 2015, there were about 200 million reported cases. Depending on the statistic, it's 200 to 400 million, but who cares about a few hundred million more, with about 500,000 deaths. A huge amount of money is spent trying to control or eliminate the disease, but what is particularly worrying, and again, this has been in the news very recently, and is particularly a concern in Singapore too, is that in the greater Mekong Delta, malaria has developed resistance against the last line of approved drugs against artemisinin. So there is now complete treatment failure of malaria cases, and as you know, it's only a 2-hour flight from Bangkok or here to here. So there again is an urgent need to develop new drugs. The disease is caused by the fact that the parasite invades red blood cells, grows inside red bloods, then destroys red blood cells and keeps the cycle going and going, and that causes the disease. What we have done, as I said, we've taken advantage of the fact of the chemistry department here, which is one of the top chemistry departments in the world, and what chemists do all day long, somehow it gives them a kick, is they try to figure out how to make new molecules, new compounds, new molecules, new synthetic pathways, and many of these molecules that they make actually look a little bit like drugs or like potential drugs. So they got together and looked at all the molecules that they had synthesized over the last 10 years and created a library of these compounds which have drug-like properties. And we took this library and screened it and said which of these compounds can actually inhibit malaria growth. And this is shown here. So each dot represents one compound that we tested. So this is 1,000 and something compounds. And if you look at the green line, any compound which is above this green line has no effect on growth. Anything that is sort of sitting in between the green and the red line has some effect, but not perfect. And anything below in the red line is actually killing the parasite. So from these 1,000 and something compounds, we ended up with 46 that seemed to kill the parasite. When we then analyzed this further, we found out particularly one compound, which we just labeled A1, which seems to be particularly good at killing the malaria parasite. What was also very encouraging is that it didn't kill human cells. So these are all human cells, and we just tested them with concentrations of this drug, very high concentrations, much higher than here, and look, do they kill these human cells? And they don't kill any of them. So this is very important to show is it kills the disease, but not the human. So what we also showed is that this compound is killing drug-resistant malaria parasites. So we have here a whole range of different parasites, and they all are killed with equal efficiency, including parasites from the greater Mekong Delta, which are resistant to multiple drugs, including, as I said, artemisinin. So that, again, this tells you this is a compound which is very promising. So in addition, it also works in treating in an in vivo animal model, which is the first step to get something into the clinic. And here is just shown different concentrations of the drug. And one is no drug, is the line here, the blue one. Here we give 4 doses of drugs with increasing concentration. And as you see, as the concentration goes up, you eventually get to a point where the parasite gets completely removed. What is important here is our best drug, artemisinin, the one that is used in a clinic. Our drug actually performs better than artemisinin in this model. So clearly, this compound is a good starting point to potentially a new drug against malaria. So now we're going to a bacterial disease, enterococci, which is worked in Kimberly Klein's lab. And this is about an idea about reversing resistance. How do you get around the fact this is resistance? Are there ways you can still use a drug against which the bacteria is resistant by doing something else? And just to give you enterococci has been identified as in high priority in terms of antimicrobial resistance. So because we have vancomycin resistance around, so it's a big concern. What does enterococci actually do? They cause a number of different diseases, urinary tract infection, inflammation in the stomach. They also can cause inflammation in the muscle, and in diabetes they cause these ulcers in diabetic foot, which can actually lead to the loss of a limb. So it's not a very nice disease, and you know we have about 20,000 cases now every year of drug resistance infections, of which 1,300 die. The numbers look small, but with these things it can go very quickly up in numbers very, very fast. So what is the problems when you think about enterococci and antimicrobial resistance? There are a number of them. One is that there is an intrinsic resistance to some antibiotics. They just are resistant to start off with. But there's also acquired resistance to a last line of antibiotics like vancomycin and daptomycin. And then there's another characteristic of the bacteria which creates a challenge for drug development is that it grows in biofilms. And I mentioned biofilms a few more times. Biofilms is ultimately when microorganisms, either by themselves, not just grow together, but they secrete other things and create a shell in which they hide, and that shell or that protective layer prevents drugs to get to them easily. So they do this either by themselves or in combination with other microorganisms to create these biofilms. So in the work that the question that Kimberly's lab is trying to address is how do you overcome this by, for example, understanding the mechanism of how an antibiotic is killing the bacteria and how resistance is developing, and then maybe coming with a second helper which creates a mechanism for the drug to work effectively again. And the other area is to understand biofilms better, to allow maybe to help antibiotics by disrupting these biofilms. So here's an example of overcoming existing resistance. So what was noted is that when you have bacteria which are sensitive and you give them this drug, daptomycin, that it always accumulates in these sort of central region of the bacteria, while in sensitive parasites, so the drug is labeled in green, in resistant parasites, I'm too much of a parasitologist, sorry, bacteria, the drug seems to go somewhere else. It was also shown that another antimicrobial peptide also seems to accumulate in this central region of the cell, and that is critical for the killing. So why is it so critical for a drug to get to this, and how does resistance lead to the drug not getting there anymore? And they showed that there is a certain gene in the bacteria. If you remove this, then suddenly the drug goes again to where it's supposed to go. And this means that this bacteria, where this gene has been deleted, now becomes sensitive to the drug again. It wasn't sensitive before. So the idea is, how can you now target this enzyme or this molecule effectively so it can't work anymore, and therefore the drug can get in and kill? So this is the idea, and unfortunately it hasn't got a solution yet, so, but this is sort of a concept of how you go after this. Another very striking observation from her lab was that she noticed that, well, E. coli and Enterococcus faecialis, two bacteria, when they grow normally, they form these colonies, but when you start mixing them together, they appear to seem to help each other to grow more effectively. And what she was able to show is that by mixing them together and these growing together, they are helping each other. They're helping each other to extract certain nutrients from the environment more effectively so that they actually become faster growing. So again here, if you can somehow interfere with this helper function, you might be able to develop new therapeutics against the disease. Okay, so that was a quick, 5-minute intro to Kimberly's work. Now, Yang Liang is an assistant professor also in SBS and similar to Kimberly also in CELSI, which is Research Center of Excellence. And he's working on particularly overcoming biofilm resistance. And there we'll give you, again, a little bit of an overview of strategies they are using and the importance of biofilms. So I already mentioned this. Biofilms are forming these sort of capsule-like structures which protect the microorganisms more effectively. And biofilms contribute to about 80% of all hospital-acquired infections. They are big problems in prosthetic joints, urinary stent, dialysis catheter infections. They also are a big problem in cystic fibrosis patients, and so on. So this is a problem which often relates to things being, in pacemaker, actually things being put into a body, a stent, and the bacteria growing on there and growing biofilms, which makes them very difficult to be treated. So what Yang Liang is looking at is how can you deal with this, and how can you deal with it by reversing the ability of the bacteria to make good microfilms. So here's an example of an approach. They screened, again, molecules which disrupt the biofilm, but they don't kill the bacteria themselves. So here's an example. You have a biofilm. You treat it with a normal antibiotic. And in green, many of the bacteria survive. If you now do the treatment of the same, but you add these biofilm disruptors, you are able to completely kill the bacteria. So again, biofilms are resistant to the antibiotic. If you disrupt the biofilm, the bacteria becomes sensitive. So his lab looks at all aspects of biofilm formation. From when it's formed, how can you disrupt it? How can you interfere with how these biofilms actually distribute throughout the body? And how can you really prevent these biofilms from forming in the first place? And he does this by bringing together a whole tool set of different techniques that exist at a university to identify, and use a reiterative process of making better and better compounds that disrupt biofilms. Okay, and now I get to the last one, which is a shift. So now I'm talking about dengue, and I think dengue everyone is familiar with in Singapore. And the problems of dengue is that it comes and goes, and it is infecting a lot of people worldwide with 390 million infections each year. Most people in Singapore know about the fact that there are different serotypes, so there are different forms of dengue. There are 4 different serotypes. And what is particularly challenging in this area is that you can get an infection with one serotype, you get immune. So you get a disease, you feel a little bit ill, you might have been diagnosed as dengue, but you recover and you're fine. And then you get infected with another serotype. And what happens then is, what is this antibody-dependent enhancement of infection? It is in this situation that you have the antibodies that you generated against the first serotype do react with the second serotype, but not in the way you want. And they make actually things a lot worse. And this is when you get severe dengue, severe hemorrhagic fever, and this is often the case why people actually die of the disease. So if you think about this, this is challenging from any therapeutic approach, because you always need to think about, I need to be able to get all 4 simultaneously, because if I don't, then I might have this problem of causing actually a more severe disease than wanted. And that's one of the challenges in terms of the vaccines that are currently being tested. And there are a number of vaccines being tested currently in South America and in the Philippines, which are encouraging at some levels, but they have this potential worrisome problem about this enhancement of disease. So Julian Lescar's lab works on, can we develop a therapeutic strategy that kills all dengue in serotypes in one go? So one fits all. And he's a structural biologist, so what he has done, and with many others, is looked at the structure of the dengue virus. And this is a model of how the virus looks. There are these molecules called E proteins, and there are 90 of them, 90 dimers of them, which make the structure of the virus. And what was noticed is in this dimer, so there are in this E protein are 3 domains, and they're arranged head to tail in this dimer. And what was noticed is that domain 3 is particularly a target of antibodies which prevent infection. Ultimately, they block the virus, and they block this by preventing attachment of the virus to enter a new cell. So they looked at this, and they were able to develop a novel antibody which has the capacity to not bind just domain 3 from one serotype, but to inhibit the domain of all serotypes equally. So if you give this antibody now to a person with dengue, it doesn't matter what serotype it has, it can neutralize them all. And this antibody is now being tested in clinical trials in Singapore, and hopefully there are no hiccups, and it will be actually available at some time in the future for therapeutic use. And this is where I will now stop with just a very quick summary. I hope I've given you a sort of a bit of a hodgepodge overview of activities that happen at NTU that are really the basis for potentially future therapeutic avenues. Clearly, infectious disease doesn't just happen somewhere else. It is a problem for even developed countries like Singapore, and it's a real risk. SARS was a wake-up call, but there, the Ebola crisis recently, it's a flight away. One flight, they have to go and stop in Dubai or Doha, and then they get on the flight here, and you have it here. I talked first off at the bubonic plague. There's, at the moment, a massive outbreak of bubonic plague in Madagascar, with hundreds of people dying of the disease. Antimicrobial resistance is spreading, and it is a threat to global health. And when I talk about global health, it is everyone. You're not immune from it. And what I think here the university has to offer to is the ability to bring together all the different expertise and skills that allows us to develop new ideas of how we might tackle this crisis before it really becomes a crisis. And as I said, there's many people to thank. I've mentioned the names of the different labs and all the people that are working in these labs on these things. I'd like to thank for sharing the data, and I'd like to thank you all for being here and paying attention. Thank you very much.
Okay, thank you very much, Professor Preiser. Can we give him another round of applause? Yes, please take up your position. Now, the next segment of our program is panel discussion and Q&A. Besides Professor Preiser, I'm going to invite Professor Stefan Trelleborg. Professor Trelleborg is the director of our Singapore Center for Environmental Life Sciences Engineering, $150 million research center of excellence at NTU. So he'll be our moderator. We have two other panelists. Professor Yeo Sin Wen. He's from Lee Kong Chian School of Medicine, and of course he studies malaria. And the next panelist is Professor Kimberly Klein. She's from, again, our Singapore Center for Environmental Life Sciences Engineering. And as you know, she studies enterococci, one of the leading causes of hospital-acquired infections. Of course, I forgot to mention Professor Trelleborg's specialization is in microbial biofilm. So with that, I'd like to pass the floor to Professor Trelleborg, and all yours, sir.
Thank you so much, and again, thank you all for being here and being here late on a Friday evening. So I guess we will try to run this so it's more a Q&A session, a discussion with the audience, but I will also ask the panel members to comment perhaps on the talk and add anything to it that they think is important for us to be aware of. I'd just like to sort of say thank you to Peter for this presentation, which was absolutely excellent. Great talk on a very important topic. It's very contemporary, and it's really a real concern that we are facing these issues. So we need to take this very seriously, and I think many of us do. And I'm an environmental microbiologist, really. The center is engaged in a number of things, studying biofilms, as you heard, but also trying to understand how ecosystems operate. So AMR bacteria and AMR genes have an impact once they are formed and pick up resistance also on systems that are outside us, outside our bodies. And it is a concern for our ecosystems. We are not quite sure what it means when resistant bacteria colonize and spread, and when the resistant genes disseminate and are picked up by other microorganisms in these various ecosystems. We don't understand what it means for the functioning. And that applies to all our ecosystems, the water, the soil, the plant ecosystems, our marine coastal systems, the air we breathe, indoor air and outdoor air as an ecosystem. And all these have microorganisms, all these ecosystems have microorganisms, and they're important for the functioning of these systems. And so anything that invades and becomes established into such an ecosystem will of course then change, right, how that community operates. And we are concerned that organisms that can colonize and insert themselves and maintain themselves with different traits, different behavior, different phenotypes will alter the functioning of these ecosystems. So that's something that we also ponder about at NTU, this broad spectrum approach that we take on understanding AMR, just as Peter has articulated the wonderful new ideas of dealing with trying to get to new antibiotics that will not lead to resistance because they simply make a biofilm less functional rather than trying to sort of to kill, they disable the biofilm. So that technology obviously is being mentored and driven at the university, and also university is keen to understand what the impact is on the environment. So we need to understand the functioning, the biochemistry, the importance of our ecosystem. So this is a very broad, to me, as also an environmental microbiologist, a very broad issue that speaks to all aspects of life and all aspects of our ecosystems, in our case, our urban ecosystem. So I think it's fair to say that we are at the university sort of aware and trying to deal with this. I think we are trying to sort of fill these knowledge gaps that we have in as broad a spectrum as possible. And the examples you gave, were really very good because they cover very different approaches that we take. So that's my additional sort of just sort of comment to as I see this problem more broadly. So, fellow panel members, before we open the floor for discussion and from the audience and perhaps questions we may have, and engage Peter again, would you like to add anything in particular to this talk?
So just want to kind of add a few things that actually the problem is actually very, very complex, and it covers actually social issues as well. So one of the main drivers of actually antimicrobial resistance, both in bacteria and in other infections as well, is actually overuse of actually antibiotics. So there's always a perception that, so I'm actually a practicing physician as well. So I actually have to prescribe antibiotics. And a lot of times one of the difficulties is being able to differentiate someone that comes in that has a viral infection. So someone that comes in and has a fever, let's say a cough, could just be having a viral infection which doesn't require any antibiotics. So the ones that you want to worry about are those that actually have a pneumonia or a bacterial infections. However, currently our diagnostics, unfortunately, aren't perfect. And so there tends to be this, sometimes to kind of be a safety first from the doctors, where they said, okay, I'll prescribe antibiotics. And sometimes there's also a perception that the patients expect it, and I don't want something to go wrong. And so it's a bit more kind of a complex issue down on the ground. And in other countries, so let's say if you take a boat, or you drive over to, take a boat to Indonesia, it's 40 minutes away by ferry, you go to Bintan and Batam. You don't need a prescription to buy antibiotics. If you feel you have a fever, you just go to a shop there. They actually sell you antibiotics over the counter. So there are a lot of different drivers that actually need to be addressed. So it would not end up to be based on what you call the magic bullet or that one single molecule or drugs that would do the job. It's actually a very, very complex issue. And we haven't even discussed is that actually 80% of the world's use of antibiotics is actually in the farming industry. And if you actually go around and sample the chickens and that come out from some of our primary products, they actually harbor a lot of a lot of antibiotic resistant bacteria there. A good example is the group B strep, the fish that came from, I think last year was, and actually affected quite a lot of our population here. Now, it wasn't a so-called a drug-resistant bacteria, but that's an example of a bacteria that came out from a food source, and even our food security actually it's quite unclear. So it's a very, very complex process. And the medical side where I work in the hospital is actually only a very, very small part. Now, that's the part that gets the most attention, but actually there are a lot of other parts that need to be addressed, and that requires actually government, political, intergovernmental, actually international. So that's why it was good to see that the WHO, the World Health Organization, actually came out with a big statement saying that AMR is going to be a huge issue, because then that actually leads to actually political action, which I think is probably needed. So we're kind of small fish, I would say, but we're helping to try and solve the problem. But it's much bigger than that.
You stole my comments. So I think I'll just bring up one other twist on all of this, which is something that you hear about a lot in the news nowadays regarding microbes, which is the microbiome, which we hear about this every day. These are the communities of bacteria that live on us and in us, and generally these are really good things for us. Our microbiome is important for helping our immune systems develop and helping us break down and digest food and acquire nutrients that we can't get on our own. And I think between antibiotic resistance and the microbiome, these are the things that, the microbiology that's in the news all the time, but there's actually an interesting intersection between those. So one of the other things that the microbiome does really well for us is protects us from the bad bugs. The microbiome is generally the good bugs, and it keeps the bad bugs out. And so one thing that really didn't come up yet is the fact that a lot of us are probably right now colonized with these antimicrobial resistant organisms. And we're healthy, and it's not a problem because the numbers are kept low. They're kept in check by that normal, healthy microbiome. And so one of the sort of cruel twists is when you take certain kinds of antibiotics that can disrupt your normal microbiome and get rid of the good guys so that the bad guys and the drug-resistant microbes can now start to grow. And those are the very microbes that are resistant to the drugs that you're trying to clear with those drugs. So I think this is an interesting intersection that's really, we hear a lot about in the press nowadays, and I think it's important to think about how those fit together as well.
Thank you, Kimberly. And at this point, would we have any questions from the audience to add to this? Comments, questions that we have forgotten to cover that you think that we should be aware of, or something that you have been pondering about for a long time? So, yeah, yes.
Just now, you all have mentioned about not to take too much antibiotics, and it's no good. And so far, I read a lot from the newspaper as well, the whole world talking about it. And what are the government doing about it? Can you enlighten us?
So I think we have to divide it up into what you call community use and hospital use. So community use, so these are your polyclinics and your what you call family doctors and GPs. So there's still what they call, there are guidelines that have come up from the Ministry of Health, so saying that if the patient fulfills a certain criteria, it comes in with fever, X, Y, but has other symptoms that you think is a viral, then you're not supposed to give actually antibiotics, and you're supposed to give antibiotics for patients that fit so-called another profile. Now, having said that, having practiced on the ground, it is quite difficult. So it comes up to actually the judgment of the doctor. And like I say, a lot of times you don't have a perfect, what the medical term is called point of care test, which means if I'm seeing a patient at the clinic, he says he comes in with symptoms of cold, but I'm not sure whether he's got chest infection that requires antibiotics. I take his sputum or his blood, put it in this test, and tells me, oh, he's got a bacteria. We don't have that test yet. So it still relies quite a lot on the judgment of the GPs. In the hospital, it's a bit different. So in the hospital, so I'm an infectious disease specialist that works in the hospital as well. If you have what they call a multi-drug resistant organism, the doctors will have to refer the cases to us. And we actually restrict what users who can use the so-called the big gun antibiotics. So some of the more expensive antibiotics that work very well against the drug-resistant ones, we actually are controlled by me and my fellow specialists. So they have to call us, they have to explain to us why they want to use it, and then we have to approve it. So those are the rough kind of guidelines. So slightly different, but basically the difficulty is that we also need good diagnostic tests, which is also part of the research going on here, actually in NTU. So to be able to tell us with good degree of certainty whether we should or should not give antibiotics. Yeah.
Hello. Does antibiotic in animal for human being consumption affected human being if we were to consume it? So if it does, at what level it affected human being? Thank you.
Yeah, so I think the levels of antibiotics in the animals so far, so I'm not the expert in this is from my reading, has not shown to be that high. What it does is actually, with the antibiotic used in the animals, you get antibiotic-resistant bacteria in the animals. And then when we eat them or get exposed to them, then we are the ones that end up with what they call the multi-drug resistant bacteria. So it's not so much the level of antibiotics by the time it reaches the what they call the chickens or the pigs, so that it's not that high. But what it does is that it promotes drug-resistant bacteria in the animal itself, and then it colonizes us.
So again, the challenge is not simply the presence of the chemicals or the antibiotic itself, but it's the presence of those organisms that spread that have acquired the resistance against the antibiotics. And the use in, you maybe did say this perhaps, but the use in animal production is extraordinarily high. So numbers we had back when I was still in Europe, for the salmon production in Norway, they used up to 15 tons annually of a particular antibiotic. Sort of massive use, and that particular drug is very stable, so it would stay in the ecosystem, it would stay in the sediments. So the selection pressure remains, right, in that case.
Just to kind of add on to your question. So just by chance, anyone here works for the Indian Tourist Board or China Tourist Board? No? Okay, so in certain parts of India and China, so I'm sure it happens elsewhere, just that the data is from there, certain of the rivers and stuff actually have higher concentrations on antibiotics than we can find in the blood when we give patients what we call antibiotics. And that's near what they call major, what they call poultry farms. So some of the poultry farms in China and India, we're talking about tens of millions of chickens and stuff. I've actually been up to China once. It was quite extraordinary. That's what I'll say. So in some of those areas, I think because of the stool and everything, they use a lot of antibiotics. The chickens poop, it gets into the soil, whatever, it gets washed out. People have done studies where the antibiotic levels in some of these streams and rivers are extraordinarily high. The consequences of that, we're not sure. So
you must have many questions for Peter, surely. After his. Yes, please, at the top there.
Hi, thank you for the presentation. I don't know if this is a question for Peter or for all of you, but I was wondering, is there a way to project how long your new drug can actually stay effective once it's released to the public, and how long it takes before it develops an antimicrobial resistance to that particular drug?
Well, I think the fastest I know of was 6 months between introducing it to the market and actually resistance being high enough that they had to withdraw it. So it can happen very quickly, but this is something that is very hard to predict. So nowadays, as part of getting a drug even approved, you often check whether it introduces resistance and how long it takes to introduce resistance. So you're trying to actually establish the mechanism of resistance before you even put a drug on market, so to avoid having these somewhat nasty surprises. In the field of malaria, what you now do is you would never give a drug on its own anymore, but always in combination with another drug, so that even if you develop resistance to one, the other one still is effective. And this is to protect any individual drug from resistance development. But until these things are, very complex to some extent, and resistance may already exist in a population naturally, and you don't see it unless you start actually selecting for this with a drug. So it's very hard to give a true predictor, but there's now we've learned some of the lessons from the past and are trying to make sure that we can minimize the risk of resistance happening too quickly.
But again, right, the way we try to deal with it is to develop antibiotics that don't have that mode of action. So the killing should not be the preferred mode of action. If we can move towards antibiotics that do other things, interfere with the proliferation or make it very difficult for the bug to go into that phase of being a pathogen simply by interfering with the natural behavior of the organism. So it just doesn't do it, or dismantle the biofilm. This is the hope that we have, that our understanding of the biology of the organism and the biofilms will get us to the point where we know where the best target is to stop its behavior. And if we do that, then there is no selection for developing resistance, right? Because we're not killing anymore. So I hope that the university engagement in this field leads to identifying an increasing number of such new targets that are. Dr. you want to say something?
I can just add an example to that I think is sort of exciting. So one of the most prevalent infections in humans are urinary tract infections. They affect most women actually in their lifetime. And so the most common bacteria that causes urinary tract infections are E. coli. And we know really well how E. coli do this. They make this little hair-like structure, and it's a very exquisite interaction between that hair-like structure and the bladder wall. And this is not my work, but people have developed very small molecules, little compounds like Peter talked about, that can bind that end of that hair-like structure. The bacteria is completely fine otherwise, but it just can't bind that bladder cell wall. And so these are now going into clinical trials as well. And so the bacteria are fine, they're never going to be resistant, they're going to be washed out in the urine. And so this is actually a really exciting, potentially very impactful, new non-killing way to limit infections.
Thank you. One of the slides about enterococci, you showed a pacemaker, and I think Peter also mentioned that stents. Could you elaborate? Does it mean that when the person has a pacemaker inserted, there is a risk of infection arising from the insertion, or is it something else? And what is it about stents? Are you referring to stents placed in the heart for patients with heart diseases?
I'm going to refer this question to the clinician here. This is why I asked him to be on the panel, because I know this is he's much better qualified.
Yeah, so there is a different risk profile for each of the different stents or different what they call bioprosthesis that you insert to the body. So pacemaker, normally it's pretty safe. Now you do get infections. It should be less than 1% of all pacemakers. I think it's like if it's done in a so-called a good center, in a good hospital, it should be 0.5%. Enterococci and another bacteria called Staphylococcus aureus are the two main what they call culprits. It's it's a difficult infection to treat because, again, of the biofilm, as what I call Peter mentioned and stuff. Now, for the stents, again, it's only a small percentage, so it doesn't happen like, to everybody. Again, it's very, very small percentage. Now, sometimes we don't know why. It can be, at any time, we can actually have what they call a transient bacteremia. And what I mean by this is that even when we go to the toilet and, pass motion, the so-called transit of the stool in our gut actually can cause bacteria to actually come out into the bloodstream for a short while. And when we brush our teeth as well, we actually have a lot of bacteria in the teeth that can go into the bloodstream. Now, normally our immune system would actually clear that, but what happens sometimes, and this is what people think, is that because the stents are there, the biofilm is there, they can actually attach. And then that causes the what they call infections in the stent. So in the normal humans, our immune system, and because we don't have any foreign bodies inside, it doesn't happen. But in small percentage, so I don't want you to go away thinking that this happens to everybody. Okay, it's a very small percentage of people that have pacemakers or stents put in that actually, unfortunately, go on to develop infections. Right?
Just now we mentioned about this antibiotic for raising of cattle and poultry. Now for example, if a chicken is brought out with antibiotics, so it carries antibiotic in the body and resistance, so when you consume it, we have this resistant bacteria as well as antibiotic. But if you cook the chicken thoroughly well, does it destroy the antibiotic property as well as the drug-resistant bacteria? That means we are safe to eat that.
So if you give the microphone to the lady there. This is her area.
Yeah, I can probably comment on that in that first of all, you indicated concern that there were antimicrobial genes in the muscle itself. The chicken meat is fine because the microbes are mostly in the intestinal tract, and they're the ones that have had the antimicrobial resistance because the antibiotics are given orally. So it's all the bacteria in the intestine. So during the processing, the chicken is sacrificed, the intestine's taken out, and unfortunately in trying to do things quickly, the contents of the intestine can be on the surface of the chicken. In the more boutique style slaughterhouses where they're much more careful, there's much less contamination. But you're totally correct, if the chicken is cooked completely, the bacteria are dead and the genes will also be destroyed by the cooking.
We talked about antibiotic. I wanted to understand how about probiotic and how it reacts with immune system, and even to the biofilm and the diseases we talked about.
So if you give the microphone to the same person.
I was just looking at Kimberly that perhaps she should take it because she brought up the microbiome concept. And I might first of all make a comment about Kimberly's work that Peter presented was the fact that when there were different mixes of the microbes, they were actually helping each other be resistant to the antibiotic. So within the GI tract, there's all those microbes there, so many different ones. And are we actually, by orally taking antibiotics, having more of a problem than if the antibiotic was perhaps administered intravenously? And so it was, and I've never really thought about the fact that the combination, so we'll get to the probiotic comment in a minute.
Yeah, so just to sort of add to that, you asked about the interactions with the immune system, and so one of the reasons, I think we don't fully understand probiotics and how these work completely, but one of the things we know is that a lot of the most effective ones are good at suppressing the immune response in general, so sort of reducing inflammation, and they generally have that sort of activity. So they're not only affecting the other organisms that they're interacting with, but they can modulate the immune response of the host that where they're present.
Yep. And the probiotic can also modulate the microbiome composition so that the system is more resistant to so it's suppressing the number of organisms that can be carrying the antimicrobial genes. So some probiotics I would recommend as being helpful in getting, if you've got a healthy microbiome, then you're less prone to the organisms that are more frequently carrying these antimicrobial genes, because they're often some of the gram-negatives, they're lower in numbers if it's an effective probiotic.
Right, and perhaps the reason you asked this question, you might know this, is enterococci are one of the more commonly used probiotics out there. And it's for these capacities to modulate the immune response and affect the community structure as well.
Yeah, and I'll just add, so that we don't scare people, that probiotics are going to cause an inflammatory response. They don't. A lot of the probiotics actually down-regulate inflammation, and there are so many diseases today linked to inflammation. So it's really is ideal to down-regulate inflammation.
I'm quite curious about the treatment you have for malaria. Yeah, because I'm working with this Indonesian community where malaria is endemic. So the problem is that people do not take their medication regularly, and it's becoming more and more resistant, right, to malaria. So is there any way that you can shorten the amount of time they need to be continuously taking the medication? Yeah.
Yeah. Yes, actually, one of the key goals for new antimalarials is a one-dose treatment. And they're actually, Novartis Institute of Tropical Diseases developed an encouraging compound which is in clinical trials, which works exactly like this. One tablet, that's all you need. Because exactly compliance is a big issue. It's an issue with malaria. It's an issue with other microorganisms. People stop taking the tablets when they feel better, even though they're actually not yet cured. And this is creates really the background to a lot of resistance. So in the malaria field, the goal is to, in an ideal situation, is to give just one dose treatment, and that is all you need to take. But there are a lot of factors that would contribute to this, and it varies from one drug to the other, because it also is dependent how big the tablet needs to be.
So can I ask Peter a question now that I did wonder about during your talk? So if it is that the way antibiotics are now developed, if it is that the first several steps that you refer to with the chemistry and perhaps the new first good library and so forth, if that's handed over to the universities, if universities are increasingly developing that first phase of 3 or 4 steps, is there evidence that we do see a change in how clever the new antimicrobials are, in that in the past there has been rather sort of traditional, and they have been having one mode of action only, which is killing, and they have been tested in suspension against free living cells and all that. Do we see in fact a change towards more intelligent drugs because of that change in responsibility being handed over to the universities for the first phase?
Well, obviously I'm not going to say we're doing it worse at the university. I think what I alluded to is, and Kimberly also, it was ultimately mentioned a number of times, is understanding the fundamental biology of a disease more effectively also gives you different ways of thinking about how to effectively interfere with this disease-causing organism. As you said, in the old days it was, a chemical that kills the bacteria from growing, and that's great. But that's as I said, is relatively uncreative, but it's based on not understanding exactly what other factors you need to consider. Now we understand that, biofilms and the fact that communities interact, it's different microorganisms interact with each other to create an environment which is either conducive to resistance or less conducive. By understanding this, we can create avenues or really better concepts. Now, and I think this is where the universities are particularly good places, because it has people in there who are not necessarily interested in developing drugs, but in understanding how these systems work. And that then is very valuable information for the other person who wants to develop drugs. So yes, I think a university is an ideal environment to come up with creative solutions. Also to come up with solutions in the sort of pharmaceutical industry, it's very time-driven. You need to find something in the next 6 months, otherwise the money runs out. And it gives very little opportunity for them to explore beyond just what the core competence. And in universities, because they have not just the task to help health, but also to increase understanding, it is this basic understanding of systems which will, I think, I'm confident, provide new and creative solutions in the future that we haven't even thought of yet.
Thank you. Thank you for that. Yeah.
I have a question. Why do the drug-resistant microorganisms usually spread in the hospital, and what can be done to prevent that?
It spreads in hospital because the so-called the drug pressure is the highest in the hospital. So most bacteria. So if you think of it as if the bacteria develops what they call a genetic mutation to the like an antibiotic, normally what it does is that it's not so fit, meaning that it cannot reproduce as fast. So the other bacteria next to it actually competes for it for food. So it means that the two of them are actually fighting. So in everything else being equal, the one with the so-called drug resistance gene will actually lose out. But once you add on antibiotics that can kill the other guy, then he's actually free to take whatever resources there is. So the hospitals are the places that use the most antibiotics, unfortunately. So it's something that we have to use, because especially in the so-called the big hospitals, the tertiary hospitals, so like SGH, like NUH, where there are patients that go in for liver transplants, there are cancer patients where the immune systems are what kind of normal, so they would be the ones to very high risk to get what I call bacterial infections. So we end up using what they call a lot of the stronger antibiotics to actually treat them. So that's why you get so-called drug-resistant bacteria in the hospitals. So I just want to make one comment. Not everything has to be very complicated. So one of the most simplest things that I was involved in, which was teaching healthcare workers in Bangladesh how to clean their hands in a hospital, and it reduced the rate of infections in patients there in the surgery. So it's because just the basic concept that they didn't really understand the germ theory. So you'd be surprised that in a lot of places they actually don't even have that basic knowledge. So just something. So a lot of times it depends on where you are. So in Singapore, of course, everybody here knows we have to clean our hands and we're very clean sort of things. But in some other places of the world, a lot of the things to reduce infections and everything does not have to be super complicated. It doesn't have to involve antibiotics. It can be as simple as teaching the community and teaching the healthcare workers basic hygiene and basic infection control.
Okay, very good point. Excellent.
I'd like to know, how will we know whether we are resistant to some of the drugs? And if we are resistant to some of the drugs, are there any way to overcome them?
I think, as it was mentioned earlier, we have, at this stage in time, from a diagnostic perspective, it's quite challenging when somebody comes in with an infection to say, look, you're infected with a bacterium which is resistant to a certain antibiotic. So often we find out that there's resistance because the treatment, you don't respond to treatment, and then you shift to something else. And I guess what Tin was earlier mentioning is, and eventually there's a stage when they bring out the big guns. So you want to?
Yeah, but so in the hospital, where I work at Tan Tock Seng, they actually, so every patient that comes in, whether he or she, whether or not she has an infection, gets actually screened. So what we do is that we take a swab from the nose and we test it for drug-resistant, what they call Staphylococcus aureus. And sometimes, depending on whether the patient has been recently in the hospital, we actually do take a swab actually from the rectum to see whether we can culture out any multi-drug resistant microorganisms. Most of the time, the yield is low, but it's done as part of what I call our infection control procedures. So if the patient cultures out positive, then we actually isolate the patient so that we keep, and make sure that he doesn't spread it to other patients as well. Outside of that, normally we don't recommend it because what happens is that sometimes you can get colonized transiently. So you can actually have drug-resistant bacteria which doesn't cause any problems. Maybe you carry it for a month or two or even a week and then suddenly it goes away. Okay, and then there's no need to actually go and treat it.
Any further questions from the audience? If not, then I think that given the time, and you have been very patient, so thank you for being here and attending and staying with us this late in the evening. It's 10 to 9. I will say thank you, Peter, and thank you, my panel members, and Aspector Ken Lam.
Yes, can we give our panelists a round of applause? Now, before you go and before they go, I would like to invite our Dean of our College of Professional and Continuing Education, Professor Ting Seng Keong, to help us present a small token of appreciation to each and every one of them. All right, Prof. Ting. Thank you. Peter, sorry. Stefan. Sinwon. And of course, Kimberly. Thank you. Thank you all, and we look forward to your presence again in the next talk, which is, I think it's on the 22nd of November, if I'm not wrong. Yeah. All right, so keep posted. We actually keep you posted, and then we hope to see you again. Thanks a lot. Thank you very much.